Good morning, or good afternoon, or good night, depending on where you are. It's my pleasure to talk to you today about the flexible genomes of haloarchaea that live in hypersaline brines. So I want to start off by introducing you to our field site, which I find quite beautiful. This is the South Bay Saltworks, and it's a salt harvesting facility here in Southern California in the San Diego Bay. And so as you can see here in the top right, we have the San Diego Bay, and that's the source of the water, which is channeled through a series of evaporative ponds within the salt works. And in those ponds, the salts begin to accumulate via evapotranspiration, and eventually they reach saturation and they begin to precipitate at that point, and so they can be harvested for commercial purposes. But in the meantime, these hypersaline brines serve as a habitat for a surprisingly diverse array of microbial life, including the haloarchaea, which are the organisms that produce these characteristic carotenoid pigments that produce this beautiful red color that's common in hypersaline systems. And so this field site is actually a really good analog for ancient Mars as the planet transitioned from wet to dry, because as you can see here on the left side of the map, we have approximately seawater to hypersaline brines here on the right. And we did manage to sample over 2 field efforts. We did manage to sample an extensive gradient from seawater to hypersaline brines. And so we covered a broad gradient in 2019, and in 2020 we decided to focus actually on this part of the gradient to fill in some of these gaps. And so we sampled extensively between seawater here on the left to brines where the sodium chloride has just reached saturation and is just beginning to precipitate out as halite. And so here on the bottom, on the x-axis, if you don't know, water activity is a measure of the free water molecules in a solution. And so water activity of 1 is pure water. And down here around 0.7 is beginning to be quite hypersaline brines. And in our first field effort, we did sample all the way down to water activity of 0.4, which is well below the known water activity limit of life. And so I will talk a little bit about our results from our environmental investigation of the diversity within these brines from our 2019 effort. And this study was actually just published recently in Environmental Microbiology. Our key findings here was that this microbial succession from seawater to presumably sterile brines is actually characterized by 3 phases. Stage 1, which is between water activity of 1 and 0.7, which is already starting to be a hypersaline brine. This stage is characterized by a surge of archaeal microdiversity, and we called it microdiversity because here on the left we have an estimate of the number of unique organisms within these brines, which you can see is increasing rapidly. But on the right, we can see that the total phylogenetic diversity is actually quite low. And so this surge in the number of unique taxa is actually due to very, very fine scale genetic diversity, and so we call this microdiversity. But this first stage, which is characterized by the proliferation of haloarchaea, is followed by a stage where the increasing selection pressure due to the increasingly harsh hypersaline conditions within the brines actually begins to drive down the diversity of haloarchaea. And at this stage, the brines are dominated by only a few highly specialized halophilic taxa. At some point, the limit of life in these brines is reached, and the remaining brines we assume are sterile, or at the very least devoid of active life. And yet we do see that nonetheless we have an increase in bacterial diversity here, which we initially thought was somewhat unusual, but we did show in this paper that the increase in bacterial diversity is actually probably infall or exogenous genetic material that's coming from the surrounding environment. It's falling into the brines and is getting preserved there. So rather than a living active community, what we're seeing here is a fossil community. But I do want to focus for the remainder of my presentation on this first stage here. This is the stage in which we see a proliferation of haloarchaea, this surge of microdiversity. And so in our 2020 field effort and in our subsequent analyses, we're trying to really zoom in on that stage 1 and try to understand what are the sources of this diversity and what are the patterns of evolution that are enabling this adaptation and specialization to hypersaline brines. So we did take a metagenomics approach in this 2020 field study, and we began with using a bioinformatics pipeline that was developed by a member of my lab, Abhishek Dutta, who I'll be sure to give an extra shout out to at the end. So one of the reasons that I'm particularly interested in taking this metagenomics approach is in order to look for evidence of horizontal gene transfer within this succession from seawater to sodium chloride saturated brines. And so horizontal gene transfer is fairly well studied in haloarchaea. It's been demonstrated that many families of haloarchaea are quite promiscuous and have high rates of horizontal gene transfer and recombination events. And so I want to understand, how does horizontal gene transfer drive these patterns in diversity, the surge of diversity, if at all? So we begin by looking for some of the usual suspects. Here I have the frequency of gene transfer agent proteins identified by RAST within our metagenomes. And it didn't take exactly the shape that I was expecting. Here we see that the peak in gene transfer agent frequency actually anticipates the surge of microdiversity, of archaeal microdiversity. And so that came as a bit of a surprise to us, but we saw the exact same pattern emerge when we're looking at the frequency of mobile element proteins identified by RAST. And so this does lead us to believe that what we have is actually a increase in horizontal gene transfer early on as the conditions begin to become a little bit more extreme. But what we have then is a latent diverse community which we weren't actually sequencing, since the latent diversity might be quite rare. And so that's our hypothesis for now. But what we wanted to investigate now is, okay, so the metagenomes are showing that there is quite a bit of change between seawater environment to the hypersaline brines, but what is the function of this change? What are the functional changes? And so we're looking at the COG functions of the genes that were annotated by RAST and eggnog, and a couple of patterns emerge. The first one is that quite clearly the most common function is unknown, and so that does tell us that there may be some more work that needs to be done in order to understand what those genes are that we haven't been able to classify. But then here we did see a pattern emerge that actually quite closely corresponds to the pattern of mobile element proteins and gene transfer agents. And so this functional class is cell wall, membrane, and envelope biogenesis proteins. And So our hypothesis here is that these proteins might be the target of horizontal gene transfer, or somehow involved in horizontal gene transfer. But then we do have a third major trend that's showing up here, and that's the cog-L functional group, and that is replication, recombination, and repair. And so that might be a little bit closer to our original hypothesis. We may be seeing that this is evidence of recombination driving some of this surge in microdiversity. We can see that this maps fairly well onto microdiversity. Obviously, these analyses are still quite exploratory, and there's more work to be done there. I want to thank everybody for listening, and in particular, I want to thank my team with Oceans Across Space and Time, which has enabled this research. Oceans Across Space and Time is funded by the NASA Astrobiology program. I also want to thank all of my collaborators at Scripps, my institution, but also my collaborators at Georgia Tech and at Stanford that made this research possible. And finally, I want to thank the Jeff Bowman lab at Scripps. That's my lab. They've been really, really great listening to me talk about my research and supporting me and helping me when possible. And in particular, I do want to thank Abhishek Dutta, who developed the metagenomics pipeline that I'm working with and has been an incredible help with all of my struggles with the bioinformatics side of things. And I finally want to thank you for listening.